Today we are going to discuss our chapter number 2, reproduction in flowering plants. So basically flowering plants are angiosperms. In this chapter we are going to focus mainly on the reproduction process of angiosperms. So before we start the reproduction process, let's take a quick revision on the various parts of the flower, how are they attached and what are the particular function of the particular part of the flower. So let's see, let's label them. See, this is the bottom of a flower, which is the swollen portion. This swollen portion along with a stalk is called receptacle. Now we will make part from the innermost portion. So the innermost portion is this. The upper portion is like bulb-like structure it has. We call it stigma. Now this tube-like structure is called style, and in the bottom we have ovary. This ovary consists of ovules in which egg cells are produced. So style, stigma and ovary together they called as pistil, and the whorl of pistils are called gynoecium. This is gynoecium. Now take another part. We'll go out portion. I'll make few structures. So its number varies in different kind of varieties. So I am making 4 only here. So let's see, label them. This is anther. This is anther. And this filamentous cylinder is called, thread-like structure is called filament. Anther and filament together they call stamen. So this is stamen, and the whorls of stamen are called androece. Whorls are called androece. Right? Now come out to side again. Here we have such structure. These are petals of the flower, or we can say corolla. Together they call corolla. Now in the bottom we have green color structure is called sepal, and together sepals are called calyx. Let's see what is the function of each part of a flower. First one is gynoecium, which is female reproductive part of a flower, and androecium, which is male reproductive part of a flower. This is petal. They are bright in color, so they attract pollinators toward the flower for the process of pollination. Next is sepal or calyx, which protect the flower in the bud form. Right? When there is a stage of a primordium, then it helps the flower in the bud stage. Right? So in this we have 4 basic parts. So petal and corollas are non-essential parts. We call them as well accessory parts of the flower. But this androecium and gynoecium are going to take part in reproduction process. So these are the essential parts of a flower. Now let's see how these all parts are attached to this receptacle. You see, we call flower is a modified shoot stem, right? It's a modified stem. So it has this apical region, apical meristematic region. There we have nodes or internet conjunction nodes we have. So basically this part The gynoecium attaches from center, apical meristematic portion. Apical meristematic portion, this gynoecium arises. And the rest of all the part like androecium or sepals are arising from conjunction nodes. So now this is the elongated part. When we stretch this part, this is stretched part, then we will now let's see the structure of androecium. This is the structure of androecium. This is the anther, and the threaded structure is a filament. Now, this anther has two lobes. I tell you, anther is of a two types. Anther may be monothekish, or dithekish. There are two categories of anthers in the angiosperms. Now, the first category, monothekish, means it has single theca, or we can say lobes. Dithekish means it has two theca or lobes. See, maximum angiosperms are having this type of structure of anther, dithekish. It means they are having two lobes. But some angiosperm which falls in the category of, in the family of Malvaceae, they belongs to this category, monothekish. So Malvaceae family. Right? Next is dithekish. So we will focus on this portion, dithekish, in which anther is having two lobes. Now this is lobe 1 and this is lobe 2. This anther is having a thread-like structure which is called filament. This point of filament is called proximal point, and this point is called distal point, because this is very much close to the receptacle, to the attachment of the flower, and this is away. Distal means at the distance, means away from the point of the attachment. So this is how the anther is living. Now next is, we will see how this filament is attached to the lobes or anther in a different way in different varieties of angiosperms. Let's see. See, here the filament is attached to the anther from the base. So such attachment is called as basofixed. So this is basofixed. Now the attachment in which filament attached to the anther at the dorsal side, we call them dorsofixed. So this is dorsofixed. Now this is in which, where the adhesion, I'll tell you later on what is adhesion, where this is, here actually the adhesion occurs. So when this filament attached to this anther from this adhesion portion, we call them adenate type of fixation. So this is adenate type. And the last one is very beautiful type of fixation, that is called versatile. What happened? You see, in this type of categories, the anther is in a dangling stage. It means when a wind comes, the pollen grains, which are mature pollen grains inside the anther, they go along with the wind and do the process of pollination. So the wind pollination is easily carried out by such type of versatile attachments. Right? Now, students, in pre-fertilization event, we will learn what is microsporogenesis, what is microgametogenesis, what is macrosporogenesis, and what is macrogametogenesis. So to understand that processes, or events, we should understand what is inside the anther. How does it looks like? So for that purpose, we will see the transverse section of the anther. So let's see. TS of anther. What do you mean by transverse section? This is the lobes of the anther. Now if we cut this anther from this portion, what is inside that anther? What is there? What kind of structures inside the anther? That we are going to tell you. So when we take this transverse section of an anther, we get such type of structure. I forgot to tell you, these two lobes are attached by connective tissue. That is connective. So we call it connective. This connective tissue is actually sterile. We call the male reproductive part help in a reproduction process. But the part of this anther which is connective, through which the two lobes are attached, it is sterile one. So it does not take part in reproduction process. Let's see. Now come here. These are the two lobes, lobe 1 and lobe 2 of dithecus anther. And here we have, what is this? This is the attachment, we call it connective. This is connective. Now inside each lobe we have two microsporangiums, the location where microspores will generate. So these are microsporangiums. So let's see, from this microsporangium to this last, we have walls. We call them anther walls. These are anther walls. Now let's see what type of layers inside the anther walls. The outermost layer is having a single layer of flat cells which are called epidermis. Right? After this epidermis, we have a single layer of the cells which are radially arranged. So these cells are radially arranged after epidermis. This layer is called endothecium. This is endothecium. After endothecium, we have, sometimes we have 2, 3, sometimes 4. So 2 to 4 layers of middle layer. Middle layer. We call them middle layer. After this middle layer, we have a special type of a layer which is having pyramid type of cells which surround this microsporangium. So here we have special type of a layer, we call it tapetum. This is your microsporangium. Okay. Now epidermis, endothecium and middle layer, what they do? They help in the protection of the anther. So their main purpose is protection. Also it aids in the process of adhesion during the shedding process. Right? तो जब shedding होती है, right, तो adhesion होती है, pollen grains को बाहर निकलना है उस process के लिए, तो उसमें क्या होता है, ये help करती है layers. Next is tapetum, very important layer. What does it do? Tapetum क्या करती है? It has a specific enzyme called kinase. ये क्या करती है? ये जो आपका microsporangium है, इसको nourish करती है. So tapetum nourishes the microsporangium. In microsporangium, we have cells which are compactly arranged. They are tightly arranged cells which are going to be microspores laterally. So these cells are very tightly attached with each other. So while they grow, this is the very young stage. Now they are developing. So when they start developing, what happens? Tapetum release its enzyme, catalase. So that enzyme break the enzyme which is present in between the cells, these cells inside the microsporangium. When this catalase going to block that callose, that is callose, which is binding that cells inside the microsporangium. Now cells are away, going apart from each other, right? Because this is also developing, this is not, this is developing and it is going to be big. This portion is going to be big, microsporangium. Now inside the cells are going apart when they get mature, right? Now they are getting apart and we are getting this type of structure inside the microsporangium after that in the developing stage. These cells which are there in this microsporangium are called microspore mother cells. Microspore mother cells, right? We call it as well MMC, microspore mother cells. They are going to give us microspores. Some of these cells they degenerate and some left which are going for the further process. Now what does MMC do? MMC are diploid in nature. Right? I am making it two lines, diploid. Now MMC follows meiosis. When this MMC follows meiosis, they start deducting. Again meiosis. Now when they go 2 times under the meiotic division, we will find the cells are finally arranged in the tetrad. We call it as microspore tetrad. Microspore tetrad. So the cells which are microspore mother cells give us microspore tetrad. They are microspore but they are bind with each other, right, in a tetrad form. So this process is called microsporogenesis. We call it microsporogenesis. Now this microspore tetrads maturing, they are maturing. When they mature, the microspore which are in a tetrad form, they leave each other. And now we have this type of microspore inside the microsporangium. These microspores will further give us the gametes which are going to take part in reproduction. Now next we are taking microgametogenesis. हमें यहीं से शुरू करना है. हमारे microspores we have. Now these microspores are going to give us microgametes. Now we will left here. These are the microspores. Now take one microspore. I am taking a single microspore here. This is microspore, which we get from the microsporogenesis process. This microspore undergoes mitosis process. Now this is haploid. Right? When it goes under mitotic division, what happens? In a microspore, we will get each single microspore is going to give us 2 cells. Right? This cell is called vegetative cell and this is generative cell. The shape of this vegetative cell is irregular, but specifically this generative cells have a spindle type of cell. So this is spindle shape. This vegetative cell will function later on, but this generative cell is going to give us a gametes, microgametes, right? So because we have two cells here, so we call it bicellular, or we call it as well two-cell stage of microscope. Right? Now, next is it goes again under mitotic division. Now, vegetative cell will remain same, but this spindle type of a cell is going to divide and will give us two cells here. This stage is called three-cell stage, or we call it tricellular. Right? Now our two sperm cells are here, or microgametes. Listen, 60 to 70% of our angiosperms fall this two-cell stage, right? And rest of the angiosperms, they follow this three-cell stage, right? Now, next is, stop. We start saying this microspores, we are saying it pollen grains, which is the male gametophyte, right? Now, after this, we get our pollen grains. When these pollen grains mature, at maturity, what happens? At maturity, shedding start happens. What happens? The walls, these walls, we studied 3 walls, they start losing their water. Right? Due to dehydration, they start losing their water. Water is coming out, it starts shrinking, and finally the pollen grains comes out of that anther by the longitudinal groove, which we call line of dehiscence. So, यहाँ से line of dehiscence, right? Because यहाँ से सारा water जो है वो lose हो रहा है, and यहाँ से क्या होगा? This is line of dehiscence, which will cut out later on, and the pollen grains will come out. Right? Now, next we will see the structure of a pollen grain. So let's see the structure of pollen grain. Every pollen grain encloses a walls, right? The outer wall is called exine, which consists of sporulin. This is type of an organic material, right, which make it tough so that it can bother the climatic conditions, the hard conditions of the climate. So this is the outermost layer, exine. After exine, we have intine. This intine does not contain sporulin like that. So it contains cellulose or other materials. So what does it do? It encloses a plasma membrane. Here the plasma membrane, and here we have pollen sacs with the pollen. So these are the layers you should know. Next, in your CBSE pattern, we have some terminologies to understand. So I'm going to discuss that 4 terms with you. Very small terms we have. Here the first term is pollen bioavailability. See, every pollen has a capability to generate tube cells or the sperm cells. So pollen grains particularly attach on the stigma, that is the female reproductive part, stick on the stigma and give rise a pollen tube, and that pollen tube helps to reach the sperm cells inside the embryo sac of the flower. So that ability is here representing the pollen viability, right? Next is pollen bags. In these days, pollens are stored in the nitrogen, right? And when they are stored in the nitrogen for particular temperature, and they helps in the process of plant breeding. Next is pollen products. So when you guys go supermarket, you will get easily this type of products because they are nutritive in nature. So for as in nutrition purpose, we started taking such kind of tablets of pollen grains, right? Next is pollen energy. So
obviously term is representing itself, like pollen sometimes do some kind of allergies in a particular persons because they do not going to tolerate this pollen grain. So sometimes allergies like eczema on the skin, you will get red rashes on your skin, that can occur. Sometimes asthmatic condition arises due to this pollen grains. And next we will take about the female reproductive part, and we will see how the Microsporogenesis and macrogametogenesis occur. That we will do in the next